The overall goal of this procedure is to accurately identify the sex of day 30 pig embryos using phenol-free DNA obtained from each embryo and 2 pairs of sex-specific primers for a PCR reaction.
This method has the potential to answer various questions related to reproductive physiology in livestock, such as the effect of maternal factors on sex ratio, including uterine capacity and the metabolic state of the sow, as well as various paternal factors.
The advantage of this technique involves no toxic chemicals such as phenol chloroform or expensive columns during DNA purification. So let's get the show on the road.
Visual demonstration of this method is as important as sample preparation. The steps are easy to learn, but special care on the details is important to avoid sample cross-contamination.
Transfer embryo samples to a negative 80 degrees Celsius freezer, according to the text. Label sample tubes with the sample ID needed for the number of samples to be analyzed. Use dry ice to half fill a thermal container and insert a pre-labeled sample tube into the dry ice. While wearing winter gloves with a pair of examination gloves on top, transfer pre-chilled mortars and pestles from the negative 80 degrees Celsius freezer to a dry ice container. Next, place a frozen embryo inside the mortar on the dry ice.